My name is Michael. I am the Director of Microbiology at Impello Biosciences. And today I'm going to be discussing microbe ubiquity, how microbes are everywhere, and the myth of sterile cultivation systems. And the overview of content for this presentation is I will highlight my background briefly. I'll define some relevant terminology in the context of this presentation. We'll talk about sterile, near sterile, and non-sterile systems, both in plant cultivation and laboratory systems. We will highlight the plant phytobiome and look at the cannabis plant phytobiome. We'll go over some recent papers talking about the importance of plant-microbe interactions and how to be conscious of your plant's microbiome during management. And we'll just do some concluding remarks, and that'll be it. So my name is Michael DiLegge. I was educated at Colorado State University. I got a horticulture degree in 2018, focusing on food crop production, and then I got my master's from CSU in Rhizosphere biology. I've been employed as the Director of Microbiology at Impello Biosciences for about 2 years now, and here I get to spearhead all the R&D that Impello gets to conduct. And my main research interests are plant microbiome interactions, host-associated microbiome systems, and sustainability in agroecosystems. So some terms that I just want to define before we get into the presentation are the word ubiquitous. So again, that's just present, appearing, or found everywhere. Microbe ubiquity in this presentation is about how most, if not all, microbe species are globally ubiquitous. Sterile in this context is free from bacteria or other living microorganisms, so totally clean. And microbiota is just a fancy term for all the microbes within a habitat or on a host. And we can see on the right side of the slide that both our bodies, here showing a hand, and plants are vessels, transportation vehicles for microorganisms. So it's really hard to keep things sterile when they're living on and within us. So some examples of truly sterile systems would be clinical laboratories. This is really as sterile as we can get. For here, we have both positive and negative pressure rooms. In a positive pressure room, air can leave the room without circling back in. The positive pressure keeps contaminants from entering the room, as air is always emitting from the room. And this also allows staff to keep their subjects that they're working on free from contamination. On the contrary, a negative pressure room, those keep surrounding environments sterile or free from contaminants. And negative pressure rooms maintain a really low air pressure to allow outside air to enter the room. This traps contaminants into the negative pressure room and prevents that internal air from leaving the space inside the negative pressure room. We don't typically see these in cultivation systems, but just to kind of emphasize how difficult the sterility can be to achieve, we'll just highlight two examples of positive and negative pressure rooms. So on the left here, we have a snapshot of an in vitro fertilization lab. That's a very sensitive cell culture technique that needs to be kept clean, whereas on the right side of the room, we have a hospital waiting room. So in a waiting room, we have all kinds of people with all kinds of ailments. We want to keep those contagens or potential contagens kept in the waiting room and not into something like the ICU, where people may be really sensitive to exposure to contagens. So these systems are typically, this type of sterile system is typically only feasible in a research or clinical setting. Now we're going to dive into plant systems. We'll start with truly sterile and get down to high microbial biome systems. So first and foremost, one example of a truly sterile system in plant research would be this. I want to highlight this tool called the EcoFab, which stands for ecosystem fabrication. This system is truly sterile, but it allows the controlled addition of microorganisms in interest of studying how microbes interact with the plant. So this device is designed to control the introduction and sampling of microbes in the plant roots or on the plant, as well as plant or microbe-produced metabolites in the system. This is a great way to study plants in a laboratory with strong efforts to control the plant's natural growing environment. So this is almost as close as you can get to a sterile system in plant growth while still having outside inputs from the environment, but the user gets to control them. And here's just a flow chart of how one might set up one of these systems, starting plants in sterile tissue culture, transferring them to the EcoFab device, the optional addition of microbes, and then you would just kind of change the liquid medium as you see fit and monitor root growth and plant growth throughout the system. You can non-destructively sample this system, and then when you it is time to fully harvest and see how your plant did, you would destructively sample it, check out the microbes, check out the roots, and check out the shoots. So these are typically employed in metabolomic or molecular biology-related studies for plant metabolism, and this is about as sterile as we can get in plant research. Another example, this is a little bit less sterile, but it would be tissue culture. As we are aware, this technique is often used for clean stock production or for researchers to study plants or plant behavior in the absence of microorganisms. These assays are performed inside petri dishes or test tubes, things that can be sterile on the inside and don't really need to be opened until it's time to sample. So you need to sterilize your plants with light surfactant like bleach or rubbing alcohol prior to introducing these systems. And if you don't sterilize your plants properly, anything that could be living on the plants, as we saw in the earlier slide with all the microbes on both the hand and the plant, can actually outcompete the plants for nutrients contained in this agar medium. So that's why it's really important to keep things sterile in here, and this is a way to study the plant in isolation of microbes. Moving forward, the next tier into near sterility would be a growth chamber environment. These often contain either tissue culture vessels or potted plant experiments. These devices can provide year-round climate control, and also if mechanical issues arise on this, more often than not, they can be fixed from outside of the chamber without opening up and jeopardizing the sterility of the inside. The oxygen or the air intake and exhaust for these systems can be HEPA filtered, just adding another barrier of sterility and containment. And because this is a small room that usually the user's arms are only in or so, it reduces the foot traffic compared to a greenhouse research bay. So again, limiting the amount of microbial vessels like humans or plants to disseminate microbes in a sterile system. And then these are often used for clean stock production like tissue culture or to just really control your environment a little bit more fine-tuned than a greenhouse. The next tier away from truly sterile systems would be lots of hydroponic or deep water culture systems. I like to call these low microbial biomass systems. You won't see this in aquaponics because the fish waste is actually converted into a plant available form of nitrogen with a microbial transformation. It's a bacteria. So aquaponics don't fall into low microbial biomass systems, but aeroponics and hydroponics and systems like that do. Typically, the only microbes in this system, outside of what's coming on the grower, are applied biological inputs such as mycorrhizal or plant growth promoting bacteria. And one thing that growers usually do in these systems is employ sanitizers. I have a small icon of an ozone generator on the left and the formula for hydrogen peroxide on the right, two very common surfactants or sanitizers used in this deep water culture system or a hydroponic system, because that will limit microbial biomass, preventing biofilms and algae buildups and things like that. And often in these systems, because the microbial conversion of unavailable nutrients is not needed, these systems also often employ salt-based plant-available nutrients. So plants can uptake them readily, and so can free-living microbes that we'll talk about in just a little bit. So the opposite of everything we just covered would be a high microbial biomass system, or also called a living soil cultivation system. This is typically when growers are cultivating both the plants and the soils that they're growing in. Some benefits to these systems include high microbial diversity, and we will talk about some benefits that can bring to your plants in a couple slides. Having living soil systems also can make sure you have a balance in your soil food web, and the soil food web is a microbial food chain that helps organic matter and other decaying materials get recycled and converted into plant beneficial compounds like humic acids and fulvic acids and organic matter. So these microbial ecosystem services are much more often in higher abundances when you're in living soil systems as opposed to soilless or substrate-less cultivation systems. And also with lots of microorganisms, things like fungi can make a lot more pore space in the root zone, which allows more oxygen both to the roots and microbial communities. Because typically our pathogens are anaerobic and our beneficials are aerobic microorganisms. So More oxygen means less favorable conditions for plant pathogens to survive to some degree. So why would somebody employ this type of system when they are considering the plant's microbiome or the phytobiome in their cultivation techniques? So now I want to talk about the plant phytobiome for a while. Breaking down the definition of this is phyto, plant, biome is ecological area, and the phytobiome includes the plant host, the surrounding growing environment, as well as all organisms living within or on the plant host. And even the cannabis plant has a phytobiome. This review by the authors in the bottom right corner of this figure, they conducted a review of 122 papers and determined the most common genus level bacteria and fungi occurring in different tissues of the cannabis plant. So from the buds, seeds, leaves, petioles, stems, and the roots, these are the most occurring genera that have been published about on the cannabis plant. So we can see the blue are bacteria and the red names are fungal genera. So the cannabis plant even has its own phytobiome, and there's probably things that these authors have left out in just looking at 122 papers from different geographical areas. But what I want to emphasize here is that although lots of growers consider sterile systems, the cannabis plant does want to have symbiotic microorganisms on top and inside of it. Now we're just going to do a quick overview of major plant endophytes. And primarily these are going to be bacteria and fungi. The first group I want to cover are mycorrhizal fungi. So these can be either ectomycorrhizae or arbuscular mycorrhizae. Ectomycorrhizae typically form relationships with woody plants, whereas the arbuscular mycorrhizae fungi are non-woody plants, so flowering plants and ferns, things like that. The biggest difference in the context of this presentation is that the ectomycorrhizae live outside of the roots and kind of form like a shield, whereas the arbuscular mycorrhizae can enter root cells, also sometimes called endomycorrhizae. And pretty much all plant symbiotic fungi can help with phosphorus uptake, especially the mycorrhizae. Second to that would be non-mycorrhizal fungal endophytes. So these things are typically found to live in or on your plants. And either the function is undetermined, but mostly these things are non-pathogenic and likely not beneficial either, just kind of commensal things that live on your plant. So all this red text are fungal genera from the cannabis plant slide that have been found to live on different organs or tissues of the plant. Whether or not these things are commensal or beneficial is still undetermined as the literature evolves. Moving forward to bacteria, everybody's likely heard of Rhizobium bacteria. So these are bacteria that can fix nitrogen from the atmosphere, allowing it to become a plant available form of nitrogen. These bacteria make homes in the roots of plants, and the genus or the family of plants that these bacteria can form relationships with is Fabaceae, or the legume plant family. So they make little houses in the form of root nodules in the roots, and they convert atmospheric nitrogen from the atmosphere into the plant available form, allowing the plant to take nitrogen up from the atmosphere surrounding it. And then non-nodulating bacteria are typically referred to as plant growth promoting bacteria or plant growth promoting rhizobacteria, depending on if they're present on the plant or in the roots of the rhizosphere system. And non-nodulating bacteria can help with mineral nutrient uptake, things like iron conversion and phosphorus conversion. They can produce plant hormones like auxin, which increases the root surface area of the plants, allowing the plants to have stronger ties to the earth and structural support, and then giving more surface area to these beneficial microbes we keep talking about. They can also induce plant host resistance genes and compete directly with phytopathogens, and we'll go into some examples of that in a few slides. But while there are a bunch of beneficial bacteria and fungi associated with plants, we can't discount the pathogenic organisms. So we'll broadly cover some pathogenic microbes. Pathogenic microbes can best be defined as those that cause plant disease. Another word for these would be phytopathogens, phytoplant pathogen disease causing. Some examples of common genus or genera of plant disease causing microbes would be Fusarium, Alternaria, Xanthomonas, Botrytis, a lot of those nasty microbes you hear about. On the left side of this slide, we see a plant infected with Fusarium. Fusarium oxysporum wilt is clogging the vascular system of this plant in an IPM study. Phytopathogens can also become advantageous when the host plant is weak or whether it's living in an imbalanced microbial community system. These are called opportunistic pathogens. So when a plant is suffering from drought stress, it can be easier for certain fungi or bacteria to infect the plant when it's trying to recover from an alternative stress. So they're opportunistic pathogens because they use the weakened plant as an opportunity to infect it more better than it would if the plant was in a healthy system. We can't talk about pathogens without talking about disease alleviation methods, and some 3 common ones would be chemical treatments, so chemical fungicides or antibiotics. However, these can also hurt your beneficial microbial community, so use those with caution. Another cool way is that neighboring plant growth promoting bacteria in the system can help inhibit the growth of phytopathogens. We see here a petri dish from the Impello lab with 3 colonies of bacillus in the top, the left, and the bottom right, inhibiting the growth of the fungus Fusarium oxysporum, which is protruding from the center of this petri dish. So where you see the smears, kind of where the T symbols are, we can see the inhibition of fungal mycelium growth by the production of antifungal peptides of Bacillus genus bacteria. So just wanted to have a visual aid for how these microbes can talk to one another and how some beneficial bacteria can keep pathogenic fungi at bay. And yeah, another way to get pathogens out of your systems is to restore balance in soil microbial communities, and we can talk about some ways to do that later in the presentation. Now I want to emphasize balancing the phytobiome and why it's so important for plant health, growth, and development. So this paper by Pash and He that came out in 2021 emphasized some key points, one being that any changes in a plant's natural microbiota, its microbial communities associated with it in the roots and on the plant body, can either cause positive or negative effects in the plant health. Homeostasis is one of the ideal situations for your plant health. This can also be called eubiosis, and this prevents community imbalances. In the presence of beneficial microbes can assist the plant in growth and development through some of the functions we've talked about a couple of slides ago, as well as how these microbes can directly compete with pathogens that can cause disease just for resources, whether or not they're preventing pathogens from infecting the plant, they're just outcompeting the pathogens for things like carbon in the Rhizosphere, allowing the phytopathogens to become unsuccessful in causing disease because they're just not getting enough food to grow in high enough cell numbers to infect your plant. Zooming in on this figure a little bit, I just want to break these 3 terms down and define them a little bit more in the context of this presentation. This is also from the Pash and He paper. So microbial dysbiosis is usually resulting from a disruption in the microbiome caused by stress or external factors, and this can be associated with host disease or making a plant more vulnerable. So an example of this in a field system could look like hail happening, causing a wound on the plant, and then an opportunistic pathogen infecting that wound for a plant. Microbial eubiosis, also called homeostasis, is a balance or equilibrium between different elements in the system, so the plant host and then positive or negative plant benefiting microbes are also kind of in balance with one another. And then this paper also defined a new term called malariobiosis, which is after a disruption is caused in the homeostasis of the microbiome, things shift in such a way to cause positive effects on the plant host under stress or disease. So one example of this could look like halophyte or salt-tolerating microorganisms colonizing the root zone to allow the plants to survive better in high saline soils. And usually soils can become higher in salts when using salt-based fertilizers, and the water evaporates, leaving the salts behind. So here we are just going to highlight 4 instances, 4 different studies about plants wanting microbiota, just some evidence to suggest that plants are actively trying to recruit their microbial communities, and how When they're not able to do that, their growth can be a little bit stunted in systems that don't have a good selection pool of microbes. So this study here came out in 2013, and it wanted to see if using a different nutrient solution as opposed to your typical salt-based nutrients in a hydroponic system was helpful at preventing disease after inoculating Fusarium oxysporum. So their nutrient solution just consisted of more organic matter, and they refer to it as a multiple parallel mineralization system. Their testing crops in this study were lettuce and tomato, and they were able to show that the disease-causing organism, Fusarium oxysporum, could be present in the system, but was unable to cause any diseases on the plants. And when other microbes were present, the total number of living cells of Fusarium was much reduced. You can see in the upper right corner of this slide that in the inorganic typical hydroponic nutrient system, there was a significantly higher amount of Fusarium colony-forming units per milliliter of nutrient solution as opposed to the MPM or multiple parallel mineralization system. So the main takeaway from this study is that it's possible for plants and pathogens to coexist in a system when other microbiota are able to be present and suppress populations of disease-causing organisms. Moving forward, another example of how plants want microbiota, the second study I want to highlight was by Chaparro et al. And they basically wanted to see if plant-deposited sugars, also called root exudates, changed over development with the plants, so through vegetative and seedling, flowering growth. And then they also wanted to see how these changes in compounds could affect the microbiota or the microbes in the soil. So they used Arabidopsis thaliana, which is just like a wild mustard, but also like the lab rat of plant research. And they showed that root exudates changed over time with sugars containing microbial accessible carbon deposited high and in higher numbers during early growth, and then phenolics and amino acid compounds were deposited higher in later growth stages. They also showed that microbes responded to these changes in root exudates by increasing the presence of functional genes that could use these compounds as microbe food. So basically, when there was more sugar, there was more microbes in the soil that could eat the sugar and grow. When there were more phenolics and amino acids, the same thing happened. More microbes that could metabolize those compounds for energy were growing. So the main takeaways from this study are that plant-deposited compounds can change over growth and development, and these are likely to accommodate the developmental needs of the plant. Microbes in surrounding soils absolutely are responding to these changes in compounds and likely colonizing the root zones. And speculatively, plants are depositing these exudates in efforts to recruit their microbial communities. Another study, also using Arabidopsis and another wild mustard called Medicago trunculata, were used to examine the relationship between those two plants and their fungal microbiome in an agroecosystem where these crops are grown every year. The authors of this study showed that when these plants were taken out of their native agroecosystem soils, they could maintain the soil fungal populations, the same type of fungi in their native soils as to when they were potted up in the greenhouse. They speculated that that was from the root exudates of these plants. So they also generated root exudates in sterile systems like the tissue culture we saw earlier, collected those exudates, and then applied those exudates to the soils in the absence of the plant. And they were able to demonstrate that these same fungi were able to grow and proliferate in cell numbers when these root exudates were applied in the absence of the plant. So the main takeaways here are that plant-microbe symbioses are likely initiated by root exudates of the host plant. And the very cool takeaway here is that the fungi were able to grow when fed root exudates in absence of the plants, because there are forms of microbially accessible carbon contained in root exudates. Lastly, I talked earlier about how certain microbes can help plants survive and grow better under times of abiotic stress. And so I want to highlight an ongoing study from Impello's research and development. In this study, we wanted to see if plants could recruit microbes in response to stress, and if these microbes were able to assist plants in growth and development under stress. We showed that we grew 2 generations of hemp and tomato plants under drought stress in the first generation in living soil systems. We then collected the resulting soils in the roots of generation 1 and applied those to a second generation of the same plant species. After the study was over, we saw that the first generation plants were able to survive the drought stress, likely due to colonizing microbes. And then we saw in the second generation that when they received microbes from the first generation, plants in the second generation had significantly increased biomass compared to plants under severe drought that did not get microbes. So on the right side here, we see the blue bars are plants from hemp and tomato that did not get microbes inoculated from generation 1, and the tan bars are ones that did get inoculated microbes from the plants in generation 1, and they were able to perform significantly better when using the generation 1 microbes that were recruited under drought stress. One caveat I really want to mention with talking, with knocking sterile systems as much as I am in this presentation is that we should all still keep cleaning, especially our indoor cultivation systems. Anytime you bring something living outside, it's going to come with its own microbes and free living microbes that are going to cause contaminants and problems. So while it is good to have lots of microbial diversity, it's also good to keep things that are really good at growing, like algae, in check so they don't outcompete with your plants for plant available nutrients. So cleaning is good because it can remove free living microbes. The best example of a free living microbe would be algae. When you get those green biofilms and slimes in your nutrient solutions, that's because the algae is eating the nitrogen that your plants want. Cleaning can also disrupt established microbes in such a way to allow better microbe competition to restore some type of balance if after populations are dampened with a good cleaning event, thus lowering the microbial load in the system that can help plants colonize more beneficials through root exudates, as we saw earlier. One example authors showed from a study in 2019 was that moist heat sterilization was applied to a diseased soil, which was able to reduce populations of disease-causing microorganisms. And then when plants were grown in those soils after sterilizing them or disrupting them, the newly grown plants were able to restore microbial balance in the soil, or eubiosis in the soil. So even though the disease-causing organisms were present, they were unable to achieve pathogenicity on the plants. And that was in a living soil system as opposed to like a hydroponic system. So cleaning is both good when you can do it because it can help lessen high microbial populations and allow your plants to selectively feed things to a degree to get more of a microbial balance. So some just general tips for incorporating your plant's microbiome into your management practice would be to make sure to implement good watering practice. I just lightly touched on this earlier, but both your plant roots and your microbes need oxygen too. So when you over or underwater plants, this is limiting the amount of oxygen or adding too much oxygen where things are drying out. The second would be to try to incorporate non-salt-based fertilizers for nitrogen, phosphorus, and potassium when possible. Obviously, it'll be easiest to alternate rather than switch from salts to organic materials immediately, but these organic materials can provide food sources in the form of microbially accessible carbon, in addition to the plant root exudates that the plants are already exuding into your system. So if you use exclusively salt-based fertilizer, this is pretty much only giving inoculated microbes in your system food from the plant root exudates only. So you want to give them a little bit more food. Some people use like molasses and home grows and things like that. Make sure to separate any biological inoculants you're using in time from applying sanitizers. So if you apply a sanitizer to your system, it's always a healthy or a good choice to reapply a biological input within 24 to 72 hours at minimum. Because these sanitizers are non-selective, they can hurt your beneficials. So don't waste your time and don't waste your money. Make sure to give some space between applying sanitizers and biologicals so you're not just sanitizing your biologicals out of the system. And then the over-application of sanitizers, most people do this to try to prevent any disease-causing organisms, can also get rid of your beneficials that your plants are trying to recruit with their root exudates. So be mindful of that. And of course, alternating chemical and biological IPM practices when possible. So for pest control or microbial pathogen control, alternating chemicals and biologicals is always a really good idea. As we just talked about, certain chemicals can also disrupt your beneficial communities. And lastly, it's always good practice to keep your grow areas tidy from any type of plant debris or loose soil or soilless cultivation debris, because certain, a piece of rock wool with nitrogen in it can grow algae and other pathogenic organisms, or other organisms that have the potential to be pathogenic. And plants that are, dead leaves that are in your system can also breed things like pathogens. So never stop cleaning, but be mindful of your plant's phytobiome when you are cleaning. So some just general takeaways are that sterile doesn't always necessarily mean good for plant growth and development, as we've seen through a bunch of examples of plant-microbe interactions covered today. Secondly, just kind of wanted to highlight some recent developments in understanding the interactions between plants and their associated microbial communities. And lastly, I hope everybody takes away a little bit more thought into beginning to or continue to consider the microbiome of your plants during management. These are my peer-reviewed literature references in order of appearance. If you guys have any further reading interest from anything covered today, especially those 4 studies I kind of sped through. And secondly, image references. And with that, that is my presentation. Thank you so much for your time and assistance. And my email address is on the first slide should you have any questions. Feel free to email me. Thank you.